kind of day? Thursday. Today's Matt. I thought it was Kay too, and so did Jeff. I told him that.
I had that in your lecture too.
So this diagram here shows the two organs that we're going to finish up lecture with, the liver and the pancreas. Like the salivary glands, those are considered to be accessory organs, and not because we don't need them. You can't live without either of them, but because food doesn't actually pass into those organs. They grow out of the digestive tube, the embryonic digestive tube. They're outpouchings from the gut. That's why they're still linked to it via ducts. But so food doesn't go up into the liver and then come back down to the digestive tract. So we're going to look at the liver first. And we kind of discussed structures of the liver when we talked about the blood supply and so on. So now we're going to come back to that point in time. The liver is the largest gland within the body, the skin being the largest gland of the body. And when we look at this anterior view, it's divided into a right lobe and a left lobe. And there is a fissure, if you will, here in the center that contains another mesentery. You can see that mesentery shown right here. It comes off the visceral peritoneum as visceral peritoneum comes up and is continuous with the parietal peritoneum on the deep surface of the anterior abdominal wall. And so let's draw that in green. And it comes down to and ends at the umbilicus. So this mesenteric structure is known as the falciform ligament. And it contains within it, disappearing onto the inferior posterior surface of the liver, the collapsed umbilical vein. You can kind of see that almost as a cord-like structure right here. And you can see it on this surface view of our liver models. But remember what this structure is right here? What we call the collapsed non-functional umbilical vein after the umbilical cord is cut. Ligamentum. It means round ligament. No.
It's a couple of them. One of them.
Ligamentum teres.
Yeah, it wasn't close.
Now, although it means round ligament, typically you see it referred to as ligamentum teres instead of the round ligament like we see with the female reproductive system. So this is the collapsed umbilical vein within that falciform ligament. Now remember just a few minutes ago when we were talking about the blood capillaries versus the lymphatic capillary system in the intestines, and amino acids and simple sugars were carried to the liver via the portal vein, and the lipids were carried via the hepatic artery primarily. In the fetal situation, all of those are going to be carried by the umbilical vein because they're coming from the placenta. So we only needed that one single vessel to carry all of those components. But once the umbilical cord is cut, that is removed. So I'm going to kind of roll this over so that we're looking at the inferior posterior surface. And we're going to look at the aspect of the gallbladder here. So we'll draw in the ligamentum teres. So we can see that on the posterior aspect. And then there is another lobe here bordered by the gallbladder. And then we have the collapsed ductus venosus up at the superior aspect, which would be the ligamentum venosum. So that is this structure right here, the white band that you see here. So that is what would have been the open or patent ductus venosus. And then the other border to that particular lobe is the inferior vena cava. So we have these two smaller lobes that we see on the posterior surface of the liver. This one's going down here a little bit long. So this would be our left lobe again, the smaller left lobe, the larger right lobe. And then we have caudate and quadrate lobes. They both have 4 sides, so that could be a little confusing. But associated with the inferior vena cava, which is against your backbone, if you will, so that's lower towards the tail. So this is known as the caudate lobe. And this one is known as the quadrate. So associated with the inferior vena cava, we have the caudate. Associated with the gallbladder, we have the quadrate. So on this real liver here, this is the gallbladder here. So here's the caudate lobe, here's the quadrate lobe. Here's the falciform ligament with ligamentum teres, and ligamentum venosa would be going up through here. The next image is a smaller liver model than what I have on this like that. And it's almost more at a right angles here, but again, inferior vena cava, ligamentum venosa would be here, caudate lobe, gallbladder, ligamentum teres, and quadrate lobe. Now, remember the ligamentum teres, as the umbilical vein, is going to be bringing oxygen, nutrient-rich blood to this central region of the liver, and that's also where the portal vein and hepatic artery are going in. All right, so that leads us to the vascular pattern and cellular structure of the liver. Go ahead and take a picture of what I have on the board, because I'm going to be drawing some cellular information here. And when you look at this particular large model of the liver, it's a bit confusing because the stand is kind of sticking out at you, like we have some large tumor on the liver. This is a bit large for my body. The liver should not extend beyond the lower ribs for more than an inch. It would be considered enlarged if you palpated it more than that, and this definitely would. Look at my entire diaphragmatic space. But that would be its anatomical position with the inferior vena cava and caudate lobe facing posteriorly. So when you look up like this, you can see the gallbladder is going to be hanging down a little bit. And then this looks a bit complicated with all the blood flow, but if you pay attention, the larger purple vessel is the portal vein. Associated with it is the hepatic artery. And this cut green structure here is the common bile duct. So within the liver, it's a common pattern. You see those 3 structures: portal vein branches, hepatic artery branches, and bile ducts. And those are known, those 3 structures grouped together are known as a portal triad. Blood flow through the liver is important and extensive. So the liver has a high absorptive capacity, and it's being our largest gland. It's going to be synthesizing our plasma proteins. It's going to be such as transferrin to transport iron, albumin, which is our primary osmotic protein, clotting factors. It's a storage site for glucose and other simple sugars as glycogen. So it needs to be taking those up out of the body and releasing them. It's a storage site for fat-soluble vitamins. So there's a lot going on across between the blood and our liver cells. Our liver cells, this is a gland, it's an outgrowth of the digestive gut. So our liver functional cells are epithelial cells. They're not simple columnar or simple squamous, they look cuboidal, but they're called glandular cells because they're not a covering and they're not a lining epithelial tissue. They're a form of a glandular cell. They're organized into six-sided lobules. Here we're looking at the surface of a lobule, all right? But we're talking, if you were to take, I know bicycle tires, wheels would work very well with six sides instead of a circle, but imagine they had six sides, with this hub in the middle and the spokes going out, and you stacked about 20 of them on top of each other. You would have this similar six-sided lobule that we see in the liver. In the pig liver, there's quite a bit of connective tissue between each lobule. So it makes it a little bit easier to pick out those lobule structures. So that you can see the six sides. Now, at the corner of each lobule, they share these portal triads. They share these three structures. So here's kind of an old diagram of the lobule, but this would be 1/6 of the side, this would be one side, and a wedge has been cut out so that we can see into the center of the lobule. So this vessel that you see right here is the same as this vessel right here. Okay? So our branches, our portal triad structures, our branch of the portal vein, our branch of the hepatic artery, and a bile duct are going to be found in each of these corners. From those vessels, talking specifically now just about the blood vessels, we have horizontal branches going out along the sides of our hexagonal structure, and then from those branches go towards the center of our lobule. Here, the branches of the portal vein and the hepatic artery join together, and the fluid content, the blood content, flows together. So we would have a branch of the portal vein, and over here would be another branch of the portal vein. And these may mix here in the center. But kind of looking on top, they're going to give branches out here that are flowing towards our central vein. So coming in from these six sides, we have branches flowing towards the central vein. Remember, we have hepatic artery components traveling up here as well. And they give branches whose blood empties into the brand new red pen. And then I probably should have purple. But this blood flow then from all six sides flows towards the center. Carrying both arterial blood and portal venous blood. So from what I said earlier, you can recognize that this blood is going to be now rich in both amino acids and simple sugars and lipids because it's got a union of both of these vessels. So we have portal vein and hepatic artery. And then these vessels that they flow into are known as sinusoids. So they are large capillaries. They have just a simple squamous wall, but they have large capillaries, and blood flows into common central vein that runs up the center of a lobule. So each of the corners I've shown is one of these. This is our portal triad. So hepatic arterial blood, portal venous blood flows into these sinusoids towards the central vein. Along the way, it flows by hepatic cells, hepatocytes. And these hepatocytes are never more than 2 cells thick. They are plates of cells that are going to be running vertically. And so these cells are going to face this sinusoid and be able to obtain material from it. And these cells face this sinusoid. So every cell is going to face a river of blood that it has access to. Now if I draw that a little bit larger, these cells don't really have microvilli, but they have what are called microrugae. They're like folds that increase their surface area. So that would be the hepatocyte. And then they face towards the sinusoid, which has a simple squamous epithelium. This is going to contain plasma. And this gap, so nutrients are going to cross this way, and the hepatocyte is going to secrete in the opposite direction whatever it's synthesizing. This gap in between the two is known as the space of Dissay. So there would be another cell right beside it. And there would be another cell backing up to it, facing the blood supply of another sinusoid. However, between these 2 cells, there are junctions that are absent. So remember, columnar cells are held together by junctions, tight junctions and cellular adherence and so on, except in this area right here. There's a separation. Now, Amy, I'm going to use you if you would come up here, please. I won't draw on you, I promise. Just back up, back to me. So in my secondary curvature of my lumbar curvature and Amy's, there's a gap there. My back's touching, our butts are touching, and there's this gap. That's, thank you very much, that's that circle that I drew right there, a small, short channel. Now, if 50 of us lined up back to back and bottom to bottom, we would have a very long channel that passed through us. That is where bile is secreted. The bile salts that we'll talk about on Thursday. And so when these line up, they create small bile canals known as canaliculi. So this is a bile. Remember the term canaliculus from bone? It's spelled the same way. And that empties into a bile duct, hence our portal triad. The portal vein, the hepatic artery, and the bile duct. It can't go into the blood because it's going in the opposite direction. Right? So back to this diagram again. Hepatic arterial blood and portal venous blood is flowing in the sinusoids by these cells, the hepatocytes. And then, not shown here, but the bile canaliculi are flowing in the opposite direction. So here's showing a bile canaliculus.
So that be a counter-current relationship?
Yeah, you could say that. It's not functionally countercurrent, but certainly in the flow pattern it is. The bile canaliculi cells are lined with, are formed of simple cuboidal epithelia. So when you look in a portal triad in the microscope, our blood vessels are going to be lined with simple squamous. The bile duct is lined with simple cuboidal. So it makes it a little bit easier to pick out. The hepatic artery is going to have a smaller lumen with a thicker tunica media. The portal vein is going to look more collapsed. In an unusual location for these particular cells, in the lumen of the sinusoids, we have macrophages. Now, we typically find macrophages in the loose irregular connective tissue, right, or in the lymphatic system. We saw an unusual place just under the surfactant in our alveoli. Those were called alveolar macrophages. These hepatic macrophages are given the name Cooper cells. They're found moving along the walls of the sinusoids. And their primary role is they're phagocytic. They break down aged red blood cells. The plasma membranes are destabilized. And part of the function of the liver is to process the breakdown products of the red blood cells, releasing the pigment, the bilirubin, and that goes into the bile along with the bile salts. So when you have liver damage or hepatic disease, there's a buildup of bilirubin levels. And the jaundice, we talked about this with newborn jaundice when we were talking about the breakdown of fetal hemoglobin. So here's another diagram showing the corners with the portal triads, the sinusoids, the central vein. And the central veins are going to empty into larger veins until they finally leave the liver into the inferior vena cava. Anybody remember what those veins are called? They're the top of the liver.
The hepatic vein?
Hepatic veins. Okay? So we just left it at portal veins and hepatic veins when we did our cardiovascular system. But this is why it's a portal system. So where would our first capillary bed be of this portal system?
In the liver?
In the liver, that's the second capillary. Sinusoids are the second capillary bed.
Is that the small intestine?
Yes, in the small intestine. So the capillaries of the lacteal would be the first capillary bed. Then it enters into the superior mesenteric vein, the portal vein, and the sinusoids would be the second capillary bed for getting rid of material secretion and absorption without having to go by the heart first. This is the model that we have of the liver histology. It's rather heavy, I don't know, 20 pounds or so. Don't drop it on your toe, it will break it. Well, the model or your toe, one or the other, not both. So this is showing not the top of a liver lobule, but kind of a wedge cut through the side, through the corner. So we're going from portal triad area to central vein area.